Technologies session indicated on the screen. Good morning, everyone. Glad to see you this morning. How's everybody doing? Good. How is anemia week so far? Okay. feeling the pallor. Okay. Well, I hopefully it's not too painful, and we'll elaborate on some of that today. Good morning. Okay, so we'll focus on iron metabolism today. So we'll talk about different forms of metabolism related to iron deficiency and conditions of apparent iron deficiency. And we'll also talk about the flip side of that, hemochromatosis. So we'll be not exclusively anemia here. We'll also talk about hemochromatosis, which is not an anemia, but it's kind of a contrast to an anemia, if you will. Okay, should we do a little review first? Okay. The turning point was causing trouble earlier, but it looks like it's okay. This is the last step in heme synthesis, right? Okay, good. Yep, so ferroketolase. So ferro, that suggests it's using iron as a substrate. Ferrous iron, is that +2 or +3? Doesn't matter. It took me a while to figure this out, but okay, if you, the S in ferrous, that's kind of like a backwards 2. And the C in ferric, that's kind of like 2 3s stacked up on top of each other and maybe backwards. Okay, so there you go. That's how I do it. No charge. Okay, very good. Okay, I think best answer here is 6, and you have to kind of think 3-dimensionally. So that iron, remember, is going to be in the center of the heme, that tetrapyrrole ring, and it's going to be stabilized by interacting with 4 nitrogens kind of in that plane of the heme. But then when it's in hemoglobin, it's also going to have something underneath it and on top of it. And those are histidine amino acid side chains that are provided by the hemoglobin protein. Okay. And if anybody get as far as physical chemistry in undergrad? Okay, so there's probably like valence structures of iron that would relate to that, right? Like 6 things that are coming out of it. Okay, all right. Okay, good. So just as a general principle, I think that stands true. So we've talked about some of the microcytic anemias, like with folate deficiency and B12 deficiency. Um, and today, with the iron deficiency anemias, those will be the microcytic. Or yeah, we talked about macrocytic anemias, and we'll talk about microcytic anemias today. Okay. Okay. Critical question of the day. All right. Henry Fenton was the one who first characterized the generation of hydroxyl radical from hydrogen peroxide, and that reaction bears his name to this day. So the Fenton reaction is really why we make such a big deal within our body about iron homeostasis. Okay, we don't have a lot of free iron or non-protein bound iron within our body, and the iron intracellularly is sequestered so that it can't be available in the ferrous form, free and floating around in the cell, to participate in this Fenton reaction to generate the most damaging and self-perpetuating form of oxidative stress, the hydroxyl radical. Okay, so this Fenton reaction, avoiding this is something that our cells devote significant attention and energy towards. So in addition to sequestration of iron, we also have other antioxidant defenses like the glutathione peroxidase and the catalase systems to eliminate hydrogen peroxide. That's another way to prevent the generation of hydroxyl radical. And then the superoxide dismutase, those three enzymes can eliminate superoxide and make the substrate for glutathione peroxidase or catalase. But if you have an abundance of iron around, or if you have an overproduction of the hydrogen peroxide, you can get the hydroxyl radical produced. So avoiding that is kind of the teleological goal of a lot of this iron metabolism that's we're going to talk about, because all cells need iron to large or small degrees, so we need it, but it also has this adverse effect or potential adverse effect. So as far as the regulation of how much iron we have in our body, the regulation of iron abundance is controlled almost exclusively at the level of uptake of iron from the diet. So there's no regulated excretion of iron. Really, it's about controlling how much of it we take up from the diet, and then in sequestering free iron or excess iron in compartments that don't make it available to participate in the Fenton reaction. Okay, so uses of iron within the body, erythropoiesis or the red blood cell compartment. If you look like statically at a person at any one time, the erythrocytes, that's where most of the iron is in the body by far. And then if you consider the iron turnover or iron flux, the erythropoiesis is, that's where most of the iron turnover is related to as well. So all cells need iron, but those erythroid precursors, as they're generating the heme iron, that's by far the greatest use of iron in the body. Okay, so if we look again at that distribution of iron, on average, males have more iron in both the erythrocytes and in the muscle than female. So let's see, we've got the hemoglobin iron. This is the erythrocyte iron that's the most abundant. Then we also have iron in muscle and in other places, and I'll elaborate on what some of those are towards the end of the lecture. This storage iron in the liver, this is the iron that's available in our body in the absence of dietary iron to support erythropoiesis up here and myoglobin up here. Okay, so the storage iron, this can go down from this to support erythropoiesis in the absence of dietary iron. Okay. And yeah, the amount of iron in circulation represented here by this transport iron, that is very tiny compared to how much is in the erythroid compartment and in the storage and muscle iron. But it's turning over very rapidly to support erythropoiesis, and we know that there are billions of red blood cells being produced every day, hour, minute. I'm going to have to remember that. But anyway, this turnover is very rapid. Okay, so just to introduce this cartoon, I don't want to walk through every aspect of this cartoon. I might just mention a few things on it, but I think this is a good summary that you might want to, have in a tab or print out and tape up on your wall as you're reviewing the rest of this and kind of trace out the different parts of it, because really everything that we're going to talk about, or most of what we'll talk about, is here. Gesundheit. Okay, so let's see. So the major compartments that we're going to talk about is absorption of iron from the intestine. We'll talk about iron's role in supporting erythropoiesis and how iron regulates gene expression in erythroid precursors. We'll talk about iron in the liver and a little bit about this storage form, hepcidin, but mainly we'll talk about the liver as being a major endocrine organ in producing a protein called hepcidin, which is the major regulator of iron homeostasis within our body. Okay, then we'll also talk about the macrophage iron and how macrophages are critical for recycling red blood cells and recovering the iron therein. Okay, so the regulation of the amount of iron is at the level of that uptake of dietary iron. Iron loss is really unregulated. So there's physiological or pathological blood loss can lead to iron loss just through, losing the red blood cells and losing the iron therein. Then we can also have, anytime you lose a cell, either from a skin cell sloughing off or gut epithelial cell being pooped out, you're going to lose some iron. But those aren't regulated processes that are going to control the amount of iron within the body. Okay, so one consequence of this is that if you get a condition of hemochromatosis or excess iron, you're going to have that excess iron until you do a phlebotomy or a chelation or something to get rid of it, because these processes are, slow. Really, when we have iron within the body, we try to recycle it as much as possible through that, going from a red blood cell to a macrophage and then back to a erythroid precursor in the red blood cell. Okay, so I guess I don't want to get ahead of myself on this cartoon, but this is a nice summary, so put a pin in it. Okay, so let's go compartment by compartment here, and we'll talk first about a simple view of uptake of dietary iron, and then a more complicated view, I guess. So the simple view is that there is a transporter called the divalent metal transporter, or DMT1, on the brush border or the luminal side of the duodenal epithelial cells. So the iron uptake is primarily in the duodenum. So this DMT1 takes iron on the epithelial side from dietary iron. It's transported into the cell. It can be used for different purposes within the gut epithelial cell, because the gut epithelial cell is going to have mitochondria and DNA metabolism. You know, it's got its own needs for iron, right? So some of the iron just stays within that enterocyte and does its thing there to support the enterocyte's needs. Most of the iron is going to be exported through a basolateral channel called ferroportin. And then ferroportin makes the iron available to the rest of the body. Okay. Now, the endocrine regulation of iron homeostasis is through that protein hepcidin. Hepcidin is produced in the liver when iron is abundant, and hepcidin acts to bind ferroportin and to cause it to be sequestered and ultimately degraded in intracellular vesicles. Okay, so this hepcidin blocking ferroportin, this is the way that we control how much dietary iron we take up. It's also a way to sequester iron inside cells in our body. So macrophages also efflux a significant amount of iron through ferroportin, and hepcidin acts on them just as it does in a gut epithelial cell. Okay, so let's look at a slightly more complicated version of this. And some of the features of iron metabolism within a gut epithelial cell are going to be common to other cells, and I'll try to highlight those commonalities as they occur. But you know, the gut epithelial cell has this special role of taking iron up within the body. Okay, so there is the substrate for that divalent metal transporter 1 is ferrous iron. Some of the iron in our diet is going to be in the ferrous form, but much of it is going to be in the ferric form because of the acidity of the GI tract. So there is an oxidoreductase at the brush border that's going to act on luminal ferric iron and reduce it to ferrous iron. The ferrous iron then is transported through the divalent metal transporter, and then as I guess it's not shown here, but some of that iron can do, as I mentioned, support respiration and DNA metabolism within the gut epithelial cell. We also, you know, if we eat meat, for example, we're going to be eating a lot of myoglobin, and we're going to be, you know, the protein of that myoglobin is going to digest away, and there's going to be heme. Some of that heme can be broken down by heme oxidases like from bacteria in the gut, and then that will release free iron that can be taken up by DMT1. But there's also a special transporter just for heme that can take that up into the gut epithelial cell, and then an intracellular heme oxidase will break that open and release the ferrous iron to join the intracellular iron pool of the gut epithelial cell. Excess iron is going to be stored in a protein called ferritin, and ferritin is that intracellular storage of ferritin that's common to gut epithelial cells, macrophages, and liver cells. So that's a common form of intracellular storage of excess iron. Okay, so in terms of that transport of the dietary iron from the lumen of the GI tract into the blood, to complete that story, the ferrous iron is going to be exported through ferroportin. Then there is kind of a reverse of the brush border enzyme oxidoreductase. An enzyme called hepcidin will oxidize the ferrous iron to the ferric form, to the +3 form. And then the +3 form, that binds to a transporter called transferrin, and that changes the transferrin to holotransferrin. I think the nomenclature, it's this free transferrin would be apotransferrin, and then when it binds to 2 molecules of the +3 iron, then it's holotransferrin. Okay, and then transferrin, this is how iron is transported from one place to another within our blood. Remember, there's no there's hopefully no or very little just free iron atoms floating around in the blood. Okay, it's all going to be hopefully bound to transferrin. Okay, let's see. So the expression of the DMT1 and ferroportin and the proteins that make up the ferritin molecule, these are under intracellular control by iron regulatory proteins in a similar fashion to what we saw with the regulation of delta aminolevulinic acid synthase from the ALAS2 gene in the erythroid precursors. So we'll revisit that translational regulation shortly. Okay, I think that's all I'm going to say about that. And just reminder that hepcidin, this is the endocrine control that is blocking this whole process in the conditions of abundant iron. Okay, so that ferritin, that's a common intracellular storage form of iron. It's also a laboratory indicator of iron status within people. I think we saw that with Isaac in our PBL case. Didn't he have elevated ferritin? I think. Okay. So now normally ferritin is going to be intracellularly, and the amount circulating within the blood should be pretty low, but in conditions of iron excess or tissue damage, you can get ferritin increasing within the blood. So ferritin in the gut epithelial cell and a macrophage and a liver cell is the same basic structure. So it looks kind of like a soccer ball. There are two genes making two proteins, the ferritin heavy chain and the ferritin light chain. And these form multimers together, making this like a hollow ball shape with multiple little pores that ferrous iron can enter. So ferrous iron can enter into those pores into ferritin. It's then converted into the ferric iron, and then it's stored as a mineral. So it's stored as a actual, I guess, like the same iron that they would dig out of the mines up in the Iron Range. Okay, so that's shown here. And yeah, normally ferritin is intracellular, and if you see abundant ferritin or excess abnormally high ferritin in the blood, then that suggests iron excess. Okay. Are there any questions? Okay. So that iron regulatory proteins, the IRP1 and 2, and I think just because everything's got to have multiple names just to make it more confusing. I think that we called this the IREBPs in last week, the iron regulatory element binding proteins. So that's another name for the IRP1 and 2. So same thing. Okay, so the iron regulatory proteins 1 and 2, these are responsible for regulating expression of ferroportin and ferritin, heavy and light chain, and DMT1 in gut epithelial cells and in other cells in a fashion that is similar to the way ALAS2 is regulated in erythroid precursor cells. So why don't we do a couple of brain teasers here. And going way back to the ECA cycle. Okay, yeah, so don't expect to have all these enzymes at the tip of your tongue, but kudos to people who selected a conatase, so that's the correct answer. That's converting or interconverting citrate and isocitrate. So I'll come back to this in a moment. Okay. And you can discuss with your neighbor if this helps. Should we, everybody select the answer that you're satisfied with here? Okay, so if you remember ALAS2, that there were iron regulatory proteins bound to hairpins on the mRNA at the 5' end. And in the absence of iron, those proteins would prevent ribosomal access to the message. And then when iron bound to the protein, it would be released and the ribosome would have access to the ALAS2 transcript, right? Okay. Okay, so let's revisit that, and that is shown here with iron regulatory protein binding to the 5' mRNA, not only of ALAS2, but also of other genes like the ferritin, the FTH and FTL. Those are the ferritin heavy and light chain genes. Then the ferroportin, that's the efflux gene. So the absence of iron is going to prevent efflux of iron from the cell, okay, and prevent that cell from storing iron. It's going to, in ferritin, it's going to make it available to the different needs of the cell. So when iron then is there's another fashion in which the iron regulatory proteins can regulate the translation of iron regulated genes. So iron response elements at the 5' end of genes, that's regulating ribosomal access to translation. There are also 3' hairpins that serve as iron response elements. And when the iron regulatory proteins bind that in iron deficient conditions, that causes the mRNA to be not degraded as fast, and that increases the translation. Okay, so the 5' in low iron conditions, the 5' iron response elements are going to be decreasing translation, and the 3' elements are going to be increasing translation. Okay, so in iron low conditions or conditions of low intracellular iron, the divalent metal transporter, that protein that would be taking iron up from the diet, the expression there is going to be increased. Okay, so you've got kind of, with respect to the gut epithelial cell, ferroportin is going to be decreasing, so you're going to not efflux iron, and the DMT1 is going to be increasing. So you're going to try to take up more iron, okay, thereby maintaining an iron homeostasis. Okay, so then in conditions of high iron, yes?
So in this picture, this is referring to the endothelial cells, right, of the gut?
This is kind of all cells at once. So I've got the ALAS2 up there too, and that would be mostly, that's relevant to the erythroid precursors.
Okay, but this is, so the hepcidin, that's a completely different protein, right?
Yes, you're absolutely correct. So this is referring to intracellular control of iron metabolism genes based on intracellular amounts of iron.
Okay.
Hepcidin is endocrine control of iron homeostasis based on the whole body's iron stores, and we'll spend considerable amount of time talking about hepcidin.
Okay.
Good question though. Any other questions? Okay, so when we have conditions of high iron, the iron is binding to the iron regulatory proteins 1 and 2, and it causes them to be displaced, and then that whole regulatory system is reversed. So you get ribosomal access to the genes that had the 3' iron, iron response elements, And then you get instability of the mRNAs for the genes that had the 5' iron response elements. Okay, so does that teleologically make sense? Okay, so where does aconitase fit into this? This is kind of a, triviality, but it's kind of interesting. The iron regulatory protein 1, IRP1, that functions in addition to being this regulator of translation, it also functions when iron is abundant, it functions as a cytosolic aconitase. So it's not the TCA cycle aconitase, but it's a same enzymatic function in the cytosol. So when iron is high, it's binding to the IRP1 and it makes an iron sulfur cluster in the catalytic site, and then that activates that cytosolic aconitase activity. So kind of just an interesting example of efficiency within the cell by getting two functions from the same gene. Okay. Let's see. So this, I think, is just another way to look at that information that I had on the other slide. So I'll just leave this for you all to chew on your own. Okay, so where are we? Back to the gut epithelial cell. So we had ferric iron being converted to ferrous iron and going through DMT1, and then heme iron going through the heme transporter and heme oxidase releasing ferrous iron going through ferroportin, and then hepcidin, this is that copper-dependent oxidase, is converting the ferrous iron to ferric iron. So I think we're at right here in our story. So we know that the body doesn't like to have free iron around, so that ferric iron, when it is generated by hepcidin, it is going to rapidly bind a protein in the blood called transferrin. Transferrin is a glycoprotein that's made by the liver, and the job of transferrin is to bind to the ferric iron and transport it to various places within the bone marrow and liver and et cetera. So transferrin, we generally have a great excess of transferrin relative to the amount of iron, and there is a laboratory value that Dr. Saxena may have talked about yesterday called the transferrin saturation. Was that something she discussed? Okay, so that's basically the amount of uniron-bound transferrin to iron-bound transferrin. And typically that's going to be, I think a high of about 50% is normal, and something like that. So if you have, and that amount of transferrin saturation is reflecting the amount of iron in the body. So in conditions of iron excess or hemochromatosis, you'll have a high transferrin saturation, and in conditions of iron deficiency anemia, you'll have a low transferrin saturation. Okay, so pretty straightforward there. Okay, so the transferrin then is going to be floating around in the blood, and it's going to be identifying a cell in need of iron, and it's going to interact with it. So I'll talk first about kind of transferrin generically, like how it delivers iron to a generic cell that needs it. And then I'll talk specifically about hepatocyte and erythroid compartments and a little bit about the kidney and kind of some details of those specific compartments. Okay, so the transferrin, when it is bound to the two molecules of the ferric iron and going through the blood, it will interact with transferrin receptors on cells that need iron. And what cells need iron? All cells, right? Except, I guess, a red blood cell that's mature and not doing any more protein synthesis. Okay, so once the transferrin binds to the transferrin receptor, then we get a pretty familiar receptor-mediated endocytosis. So the receptor is endocytosed, or the plasma membrane invaginates. It forms endocytic vesicle. Then as that vesicle is acidified, the transferrin is released. The transferrin receptor then can be recycled back and re-expressed at the plasma membrane. So that then leaves the iron in the ferric state within a little endocytic vesicle within the cell that needs iron. And the transport of the iron out of that endocytic vesicle is really recapitulating the transport of dietary iron from the lumen of the gut into the gut epithelial cell. So there's an oxidoreductase called STEAP1 that is converting the ferric iron to the ferrous iron, and then that same transporter, the divalent metal transporter 1, is then exporting the ferrous iron out of the endocytic vesicle and into the cytoplasmic iron pool. Okay, so that's a common way that iron can go from the circulating form in transferrin to be available intracellularly for any cell that would need iron, a hepatocyte or an erythroid precursor or any other cell. Okay. And some cells then, like, and particularly macrophages and hepatocytes, and for different purposes, they will efflux excess iron, and that then really recapitulates again the efflux of iron from a duodenal gut epithelial cell, where you get the ferroportin exporting the 2+ iron, and then in the same enzymatic function as hepcidin in a gut epithelial cell, but it's a different enzyme called ceruloplasmin, will then convert it to the +3 form, and then it binds to transferrin and does its thing. Okay. Let's see. I think, yeah, so this is just showing that recycling of the transferrin receptor. And then some of the things that the iron can do intracellularly. It can, of course, be a substrate for the ferrochelatase at the last step of heme metabolism. And once heme is made, it can be converted, or it can be added to the globin portion of hemoglobin or myoglobin. There are also uses of iron in iron-sulfur clusters in the electron transport chain enzymes, and not shown here, but there are many enzymes in the nucleus, like ribonucleotide reductase and primase, and some of the DNA repair enzymes that are also dependent on iron. Okay. Okay, I think I am kind of getting ahead of myself here, but this is all really showing the same thing. Excess iron within all cells, really, I guess I think it's safe to say, is stored in those little protein balls of ferritin. And then in hepatocytes, because the liver is the storage organ for iron, they have this other form called hemosiderin, which is a kind of a bigger aggregation of the ferritins that is the labile pool of iron that can sustain iron homeostasis in the absence of dietary iron. Okay. All right, so before we go to iron more specifically and what it's doing in the erythroid cells and hepatocytes and et cetera, are there any questions? No? Yeah?
Is it the case that ferritin can only bind to ferrous iron?
Yes, so the little pore is going to be taking in ferrous iron and also spitting out ferrous iron.
Like ferritin in the serum, if it were to get into the serum, would not be able to sequester any like ferric iron in the ferric form that's like floating around or anything.
I don't think so, no. So that would be, the way that we would get rid of that would be ceruloplasmin, converting it into the ferrous, and then it binding transferrin. And, you know, in most people, there's a big excess of transferrin binding capacity So yeah, that would be how that would be sequestered. I don't think like circulating ferritin is going to be sucking up excess iron. Yeah.
Hey, going on the same line, transferrin does it only bind ferric iron?
Yes.
Okay.
Yep, that's correct. So it's depending. So this whole process is dependent on copper. So hepcidin and ceruloplasmin, those are both copper-dependent enzymes. So, you know, we need copper in just tiny, tiny amounts in the diet, and that's one of the reasons that we have it is to maintain iron homeostasis. So, yeah, that transport of iron through transferrin, that's dependent on the activities of ceruloplasmin and hepcidin, which are dependent on copper. Okay, good questions. Any other thoughts? Okay. Why don't we talk now about iron in erythroid cells? So these are cells in the erythroid lineage in the bone marrow that are going to become red blood cells, but they haven't yet lost their capacity for gene expression, right, and protein synthesis, etc. Okay, so in these cells, they're going to be taking up iron in the same fashion as other cells through that transferrin receptor-mediated endocytosis, and then the steep oxidase and the DMT1 transporting it to make the ferrous iron in the intracellular iron pool. So by far the major role of iron within an erythroid precursor cell is going to be as a substrate for ferrochelatase and for making hemoglobin. Okay, so just to review the structure of heme, the ferrous iron sits in the center of the tetrapyrrole ring, and it's stabilized by those 4 interactions with the nitrogens in the pyrrole rings, as well as, let's see, I think that, yeah, histidine on the top and below it. And it's that interaction with those histidines above and below the plane of the heme iron that provides an interesting cooperativity or cooperative relationship between the binding of oxygen on one subunit of the hemoglobin and the affinity for the other 3 subunits of hemoglobin for iron. So as the iron is binding to oxygen, it's actually the iron is interacting with a proximal histidine and then a distal histidine, and the distal histidine is down here and not shown. But when the oxygen binds to the heme iron, it's pulling the heme iron further down into the plane of the heme, and that's pulling that histidine that is on the north end of it and an entire alpha helix down further closer into the heme ring. And this has the effect of changing the shape not just of the one hemoglobin subunit that bound that oxygen, but all the other 3 subunits as well. And that is shown, I think, nicely from this Protein Database 101, which is a great website, a little illustration. So this is showing oxygen binding to one subunit of heme, and then you've got 1, 2, and back here, these are the other subunits, and you're getting pretty dramatic conformational changes in those other subunits just based on the conformational change in the one subunit. So that conformational change in these other subunits increases their affinity for binding oxygen. And that is why in the heterotetrameric hemoglobin, if you look at the oxygen saturation curve, it is sigmoidal, right? So it's got like 2 or multiple phases in it because there is the affinity of oxygen for binding is dynamic based on how much oxygen is already there, right? Now, in contrast, the myoglobin, which is essentially doing the same thing, right? It's heme iron binding oxygen, except it's in a single monomeric protein, right? So it doesn't have any capacity to influence binding of one oxygen to myoglobin doesn't have any influence on binding of any other oxygen to myoglobin, right? Because it's not in the same complex. So there, the oxygen saturation curve for the myoglobin is hyperbolic, okay? But if you have this multiple subunits as in hemoglobin, you get that sigmoidal curve. Okay. So now we're in iron in erythroid cells, and that's what it's doing as it's going from your lungs to tissues and back and forth. And we talked about some of that, I think, last week. I just want to go back a couple of slides. And I'm not sure if we've mentioned this or not, but with iron, some of the utility of iron within our body is that it can exist in stably in both +2 and +3 oxidation states. So in some cases of iron, when it's used, for example, in a cytochrome P450 enzyme or an iron sulfur cluster in an electron transport chain enzyme, you're going to get a transient change in the oxidation state of iron during the catalysis, right? It's going to go from +2 to +3 and then back during catalysis. With oxygen binding, the binding and the release of oxygen on hemoglobin, there's normally no change in the oxidation state of the iron. It stays in the ferrous form. Okay. Now the exception is, of course, with the formation of what? Methemoglobin, right? There you get, when you get a lot of oxidative stress around this oxygen, instead of leaving as diatomic oxygen, it can leave as superoxide, and then that will leave the iron as a +3. Okay? And then that's the definition of methemoglobin. Okay. All right, so let's consider now our red blood cell has our iron in it, and it's been circulating around the blood, doing its thing, delivering oxygen to tissues and taking carbon dioxide away from tissues. And then it's, over time, it's exposed to oxidative damage, and it doesn't have capacity to re-express genes or repair damage, really. So every red blood cell eventually is going to die because due to oxidative damage, right? So the major way in a healthy person, the major turnover route or the way that those aged red blood cells are eliminated is to be endocytosed by macrophages, and principally macrophages in the spleen and the reticuloendothelial system. So the macrophages will envelop and engulf a aged red blood cell, and then chew up the different constituent parts. So the major protein part is hemoglobin, and that'll be digested down into amino acids, and those will join the amino acid pool in the macrophage, and then excess amino acids will be exported out into the blood. As far as the iron from that red blood cell, the heme is released as the hemoglobin protein is digested, and then there is a heme oxidase which will release the ferrous iron into the macrophage. Now, the macrophage, I mean, it can really almost, it's just about as important as the gut epithelial cell in terms of making iron available, right? Because you've got this constant flow of making red blood cells and red blood cells being degraded through this process. So the amount of iron that is leaving the macrophages through that same mechanism really as through the gut epithelial cell, that's a huge amount of iron, right? We only need a tiny amount of iron really per day because most of it is recovered through this mechanism. Okay, let's see. So where are we now? We're going to return in 10 minutes and talk about a little bit more about how we get iron inside of a macrophage and then what happens to it afterwards. So why don't we break and come back in 10 minutes?